Welcome back to BioBites with Mick. In today's video, we're diving into an exciting area where artificial intelligence meets the cutting edge of cell therapies, AI-guided receptor engineering. Hey there, in previous videos, we've explored various aspects of biology and the computational tools, particularly AI, that are being used to engineer better receptors for cell therapies, ultimately aiming to improve their performance. So what is the big deal with receptors in cell therapies? Well, in therapies like CAR T cells, engineered receptors are crucial for ensuring both safety and efficiency. These receptors need to remain inactive until they encounter a specific disease target, like, for example, a tumor cell. However, a major challenge is that even subtle structural changes in these receptors can lead to significant functional consequences. Unfortunately, the structure of these engineered receptors is often poorly understood, which can negatively impact how well therapy works in the first place. Think about it. If these receptors are active even when there's no target around, which is a phenomenon called tonic signaling, it can lead to T-cell exhaustion and ultimately reduce the therapy's ability to fight the disease. This is where improved methods for tuning receptor functions at a structural level are desperately needed. This brings us to a fascinating study from Northwestern University, where they presented a new structure-based analysis to guide the design of synthetic receptors with improved performance for cell therapies. So their work explores the structure-function relationship in these engineered receptors using computational structure prediction. The researchers hypothesized that structure prediction might be most effective when working with data from natural receptors. Given that current prediction tools often rely on comparing sequences to known proteins. So to test this, they generated structural models of receptors to investigate how their structure relates to their function. They used a case study that involved converting natural human cytokine receptors into engineered ones. The study revealed that specific structural features in the receptor's ectodomain, the part that extends outside of the cell to trigger signaling, and the transmembrane domain, which spans the cell membrane, explained a considerable amount of the variation in receptor performance. Performance was measured by how much the receptor turned on a receptor gene. Beyond just the structural features, the authors looked at categorical variables, such as the specific choice of protein domains and how they were combined. This helps capture properties unique to protein chains that aren't directly described by the structure alone, like how well the protein is expressed, where it localizes in the cell, and its stability. Overall, they found that structure-function trends were largely consistent across different sets of receptors, even if their structure varied. Now, you might be wondering, how does AI come into all this structure prediction? Well, engineering these receptor cells is tough because they are relatively large and have tricky transmembrane domains, and they can often adapt to different shapes. Well, thankfully, we've seen groundbreaking progress in protein structure prediction tools like AlphaFold and Rose's TTA-fold. These AI-powered tools have been incredibly successful at constructing complete 3D models of natural single-pass transmembrane receptors based on their sequence. The developers of AlphaFold and Rose TTA-fold even shared the 2024 Nobel Prize in Chemistry for this very achievement. However, even with these powerful tools, figuring out exactly which structural features are most important for receptor performance is still a challenge. The lack of structural knowledge forces researchers to spend more time and effort empirically exploring design choices. Even when good designs are found, it's hard to understand why they work so well without this structural understanding. This is where the Northwestern University study offers valuable insights. They proposed potential design rules that can be tested experimentally and can guide future engineering efforts across different receptor families. The researchers think that these insights will be most immediately helpful with designing synthetic receptors that use similar signaling methods to natural receptors that they've studied. Looking ahead, perhaps the next step of this technology is generating libraries that explore a wider range of design possibilities. Ultimately, this study provides an optimistic view that protein structure prediction tools, while of course still improving, are already able to accelerate the development of synthetic receptors for a wide range of biomedical applications. It's really interesting to see the impact AI is having on tech, of course, but also specifically biotech. The intersection between technology, data, biology, and AI is really what I'm interested in, what the BioBytes channel is all about. So if you found this video interesting, make sure you like this video and subscribe to the BioBytes channel. Let me know in the comments below if you have any questions or topics you'd like me to cover in future videos. Thank you for watching, and I'll see you in the next one.